Welcome everyone. Here is a question from GATE 2022 Biotech exam. The question says, a circular plasmid has three different but unique restriction sites for enzymes A, B, and C. When enzymes A and B are used together, two fragments of equal size are generated. Enzyme C creates fragments of equal size only from one of the fragments generated by those cleaved by A and B. The plasmid is treated with a mixture of A, B, and C and analyzed by agarose gel electrophoresis. We need to mention the number of bands observed in the gel. As we all know, restriction enzymes are endonuclease that recognize a particular sequence on the DNA and cleave at a specific point. As mentioned in the question, the DNA here is circular plasmid. So the presence of a unique site for a given restriction enzyme will result in the generation of a single fragment. I hope this should be clear to each and every one of you. So let's assume that if there are three restriction enzymes and those are having a unique site, that will result in the generation of three fragments, 1, 2, and 3. There are few extra information which is given here, and those are when enzymes A and B are used together, two fragments of equal size are generated. So as said, A is having a single site, unique site, B is having a unique site. Now, when these restriction enzymes will cleave this circular plasmid DNA, this will result in the generation of two fragments. But since the molecular weight of these two fragments are going to be identical, we'll be only observing a single band on the agarose gel. As we all know, agarose gel separates biomolecule on the basis of molecular weight as well as on the basis of conformation. Considering that the conformation of both the two fragments are same and molecular weight is also the same, I'll be getting a single band. Now, the second part of the question says, enzyme C creates fragments of equal size only from one of the fragments generated by those cleaved by A and B. So I can consider any of these fragments, 1 or 2. And as mentioned already that all these restriction enzymes A, B, and C are carrying a unique site. So let's place C somewhere here. You can either place it on this end or this end. I'm placing it here. But the information which is given here is that even the C creates fragments of equal size. And when I'm saying equal size, because it is cleaving this fragment in between somewhere, which is resulting in the generation of the fragments which are of identical molecular weight. So whenever, even if there are 4 or 5 or 6 DNA fragments, but if all those fragments are carrying a same molecular weight, those are going to appear as a single band. So now let's complete this question. It says this plasmid is treated with a mixture of A, B, and C. So let's place these restriction enzymes on this circular plasmid. So let's keep A here, B here, and C here. Now what will happen? If I'll digest this circular plasmid DNA with A, B, and C all together, I'll get this fragment 1, 2, and 3. Remember, the molecular weight of first and second is identical. These are having a identical molecular weight as well as the similar conformation. So that will result in the generation of a single band on the agarose gel electrophoresis. So I'll get one band from these fragments 1 and 2 and the another fragment from this end, which I'm calling it as a third. So if I'm running my sample on the agarose gel, these are called as the wells, I'll get 2 fragments depending upon the molecular weight. So the third is going to be heavier. So this is corresponding to the third. And then these two are corresponding to the first and second, because agarose gel will not be able to distinguish the first and two because of the identical molecular weight as well as the conformation. However, you will see some intense band because there are 2 fragments here, and I'm placing 1 and 2 at the bottom because those are going to be of low molecular weight compared to the third fragment. So I hope this is clear to each and every one of you. The other thing I would recommend you solve this question considering that the DNA is linear and your answer will vary. I hope this solution is clear to each and every one of you. If you do have any concern, feel free to ask. You can mention your query in the comment section. Thanks for watching the video.